In this chapter 6, section 6.5, we are only going to study a specific molecular genetic technique, which is inactivating the function of specific genes in eukaryotes. That is the only molecular technique that we are studying in the cell biology class, and this will be the last part, and this will be the last part 5 of the module 1.1. So in this process, what we actually want to do or actually want to achieve is replacing the normal genes with other sequences. And in this process, the gene is modified. The second inactivation approach is introducing an allele whose encoded protein inhibits the functioning of the expressed normal protein. So here the gene is actually unmodified, but we are actually inhibiting the protein by another allele. Another way that we can also use a technique where the destruction of the mRNA transcribed from a gene is also used. Here the gene is also unmodified, but the mRNA is actually degraded, which will disrupt any protein synthesized. No protein will be synthesized from the mRNA, and the gene will appear as silent. So let's take a look at a couple of different approaches. So the first one that we are looking at is the homologous recombination with the transfected disruption construct. So what we are doing in this process, so this is actually used routinely in yeast, and you have a target yeast sequence. Now, on the target yeast gene sequence, you have a 20 nucleotide flanking sequence and a 20 nucleotide flanking sequence, and you actually design a primer, which is complementary to the primer, complementary to this flanking region and this flanking region. Just name them primer 1 and 2. And you just do a PCR reaction, and in the polymerase chain reaction, you will actually introduce something around a kanamycin MX, CAN-MX gene, which will have this same sequence on each side of it. So you are taking a sequence of your target gene sequence, and you are designing a primer, and you are using in vitro a kanamycin sequence as well in the primer. So see that this is the flanking sequence. This is a little bit of sequence of the kanamycin gene that you want to amplify. This is a little bit of sequence of the kanamycin gene you want to amplify, but you also contain this portion of the nucleotide fragments from here. So now you actually do have a disruption construct which contain a kanamycin resistant gene, which is resistant to a specific G418. We call this as a genticin. So this is a selection marker. So this is your disruption construct. So now what you do that you actually take an yeast diploid cell and you actually transform or transfect, means that you actually introduce this gene to this yeast cell. And there are chemical methods to do that, or sometimes we can just use a technique called electroporation. We just use an electrical field to pulse them. And the cell will take up the DNA, or we can do a chemical treatment where the cell will actually take up the DNA. So it's transfection is the word that we usually use for the eukaryotic cell. Transform usually we use for the bacteria, but sometimes they are actually used interchangeably. So now this disruption construct is inside that yeast cell. And as you can see that the yeast gene, which is the target gene, actually do have these flanking sequence, and the kanamycin gene also have do the same sequence in the flanking region. So this may undergo homologous recombination, and we are going to select the cells or plate the cells with this G418. So kanamycin, as I said, gives resistance to G418 genticin. So now you will see that the sporulation will happen, and there is a possibility that from this diploid yeast cell, you should get like a 4 haploid spores. So if you look at these are the two possibilities that you have the disrupted gene. That means that the gene copy right here is disrupted, whereas these are the normal. If the gene is disrupted, then this will actually grow in presence of this G418. On the other hand, what happens, if this gene is actually non-viable, these will actually not survive. So what happens that if this gene is essential, it will not survive. If this gene is not essential, then this will also survive in the selection media. So that's one of the drawbacks of this system, that if you silence the gene, you really do not precise function of that gene if you just completely, if it doesn't grow. That's the only shortcoming of this process. Other than that, you can actually utilize this process pretty much to knock out something. So how we actually do this gene knockout in the mouse germline, if you just look at this technique by this process, suppose this is a construct where you have neomycin resistance. This is also resistant to G418. And here is another gene. This is a thymidine kinase from a herpes simplex virus. Now, what you have that you have a flanking sequence very similar to the flanking sequence of your gene X that you want to actually knock out. So you actually do an homologous recombination, thinking about that these gene of interest will be actually do an homologous recombination, and here your neomycin resistant gene will be actually inserted. On the other hand, what you are seeing that this thymidine kinase gene, this is actually not inserted in this condition. So this is not inserted, this is inserted. So now this is a targeted insertion. So now if you grow these cells, the mutation in the gene X cells are resistant to G418 because this actually confers the resistance to G418, and you also grow them in presence of ganciclovir. So what happens if you have the thymidine kinase gene in here? Thymidine kinase is actually sensitive, and it will cause cytotoxicity if presence of ganciclovir. So it will not grow. So this means that this has not been in here. So this is a negative selection for the recombinant, and this is a positive selection for the recombinant. And this is a very intelligent way to form or do a knockout. On the other hand, here you can see that suppose in an embryonic stem cells, you are undergoing a process where you have a non-homologous recombination. You have a gene, and somehow this thymidine kinase gene has inserted in somewhere. So if you actually have this non-homologous recombination, then you actually have the neomycin gene right here, as well as the thymidine kinase gene. So what happens that if you have the thymidine kinase, it will be sensitive to ganciclovir and the cells will not grow. So if the cells grow in presence of G418 and ganciclovir, you can specifically tell that only the homologous recombination took place to do the target insertion into the gene of interest. So here is another way to look at it. You can see that the non-recombinant cells, they actually will not be selected. Recombinant with tandem insertion will have these thymidine kinase. On the other hand, the recombinant with a gene target insertion will only have these green part. So when you just do the both selection with G418, it will select these thymidine kinase insertion as well. But if you select, which is a negative selection with ganciclovir, only the one with this will be selected. So this is also another way to do a gene knockout in the germline. So here we are looking at the same process that in reality, that how this germline insertion actually can happen. So you take the embryonic stem cells, and you can see that we are looking at this gene X, and here right here is a thymidine kinase, and here you have the neomycin, and this is the gene X that we are interested in. And right here, this is actually the target vector. This is the targeting vector, and then the target gene right here is actually of interest. And in this process, what we are seeing that we actually only got the neomycin by the homologous recombination, but not the thymidine kinase that we have seen before. So in this process, we actually only have neomycin R will be selected. So here is a beautiful picture to show that how these embryonic stem cells are actually implanted into a mouse after doing this process in vitro or in a petri dish. You actually take these specific gene-targeted ES cells with the disruption or the targeted gene where you actually did that, and then you take a glass pipette And this is actually the blastocell. So this is the blastocyst, and this is the inner cell mass, or the ICM. And right here, this empty space is the blastocell. And using this glass pipette, you are introducing these embryonic stem cells into the inner cell mass. And the blastocyst is held by a pipette right here, so that it is just a gentle suction will keep it in here, so that you can steadily use a glass needle into the blastocell and deliver the embryonic stem cells. And then these will be actually there will be some mouse that where this will be actually implanted into the germline, and you will have a specific mouse right here. And you can see that the ES cell-derived chimeric founders, you can have all of these from the generations. Now, if you actually take, say, suppose this black mice right here, and if you just actually do a cross with this, which is the C57B L6J, which is this the black background. And at this point, if you actually just do this cross, you should actually get a ES cell-derived founder within the cross. So this is how you can actually make a transgenic with the specific gene knockout. So another way that you can actually do the transgenic, and this is by the random gene integration. This is not by the homologous recombination. You actually take a fertilized mouse egg, and prior to the male and female nuclei are actually fusing, you actually inject the foreign DNA into one of the pronuclei. So when you do this process, in this process, what you are seeing that you transfer the injected eggs from here into a foster mother. So what happens, about 10 to 30% of the offspring will contain the foreign DNA in the chromosome of their tissues in the germline. And now you breed the mice and express the foreign gene and some of them will actually be integrated into the germline. So this is also another way, which is a random gene integration by this process. So another advantage is that the last two methods that we actually talked about, if a gene is lethal, then the mouse will not survive, and really you cannot do much experiment if it is at the germline. But here there is an advantage of this, where we are trying to do a tissue-specific knockout of the somatic cell, so we can tell that a particular gene, how it functions in a particular tissue. And we'll look at the application later. So let's first take a look at how we actually do that. So here what we have, that this is a mouse which actually carry an endogenous gene. So this is a transgenic mouse this is called a LOXP mouse. So what we have in this, that we have this LOXP site, flanking the exon of number 2. So this is actually this is the exon 1, exon 2, exon 3. This is the gene of your interest. These are the introns. And in the introns, you have the LOXP in the introns. You have the LOXP in the introns. So this is basically the typical construct. So here, what happens, this cell is not expressing the CRE. So you do not have that. You have another mouse which actually has a heterozygous for the gene X knockout, and all cells carry the CRE gene. So right here, you have a cell type specific promoter, and downstream of that, you have this CRE sequence. So think about that suppose this is only activated in liver cell. So you can just put a specific liver cell specific promoter, and then the CRE will be produced only in the liver cells, not in other. If you actually put a sequence of the promoter which is neuron specific, then it will only express in the neuron. So this is how you actually do a tissue specific promoter. So now you are crossing these two mice. So what is happening, that here you have a gene function is normal, where you have an exon 1, exon 2, exon 3. When an mRNA will be produced, all these introns will be sliced out. Exon 1, 2, and 3 will actually all come together, and it will produce the functional mRNA, and that will be translated. So the gene is functional. Now what you are seeing right here, when you actually cross this, these cells CRE will actually identify the LOXP sites right here, which is in the two introns flanking the second exon right here. And it will actually cleave the second exon out. Now you have exon 1, exon 3, but the exon 2 is out. So now here you have the middle part of the gene is gone, and this will actually silence the gene, and the gene is now non-functional. So the advantage of this process is that you can use a cell or tissue-specific promoter so that the Cree will be only produced in a specific type of cell, and this knocking out of the gene will only happen in a specific cell type, not in the germline. So if the gene is essential for the survival, this will only inactivate it in certain tissues or cells, but not the whole animal, so that it will not survive. So here is an application that you have, you can read it in your book as well, is about a role of a glutamate receptor. So the scientist actually first did this germline inactivation, the process that we saw at the very beginning. So in that process, what happened, preneonatal stage, all the mouse actually died. So it was actually lethal. So then what they did, that they inactivated the gene only in the hippocampus of the brain, and they found these mice actually develop into the adult, and they do have a learning deficiency. So what this glutamate receptor does, you can see that it is in between the communication in between the two neurons, and these are the glutamate receptors which actually receive some neurotransmitters. So this is a typical structure, extrographic three-dimensional molecular structure. We have seen a lot of images of these. So this is a typical glutamate receptor. This is just emphasizing that the story, it has nothing to do with any specific information that you need from here. You do not need to know the domains or anything like that. This is just an overview, reinforce the take-home message. So basically what happened, that when tissue specifically in the hippocampus in the brain, this glutamate receptor was inactivated, it led to learning deficiency. So this was the advantage of the tissue-specific knockout of a certain gene function. So on the other hand, this is actually a little bit more complex process where we actually knock out a dominant negative allele. So here, what we are looking at in the step 1, these embryonic stem cells of the heterozygous of the gene knockout gene mutation, and we have a homozygous marker gene of dominant allele, which is the brown coat color, are transplanted into the blastocell cavity of about 4.5 day blastocyst that are homozygous to a recessive allele of the marker. So remember that the black mouse allele is a small a, X, capital X plus, and then you have capital A, capital X minus, and X plus. So this brown mouse, this is actually coming from in the injection into the blastocyst, and this is actually the blastocyst of a black mouse. So now it is surgically transferred to the foster mother. Now the surrogate, they will actually produce the offspring, and you will have the chances of either you will produce a black mouse, which is this, or you will produce a chimeric mouse. And the chimeric mouse means that it actually crossed over these genes. And if we actually select the chimeric mice for the cross, so the possible genes, remember that all of the black mouse, their allele will be A and X plus, because see that it is AA, X plus, so you will have AX plus, AX plus. So all of them are X plus in their one of the allele. So this is a back crossing. So first you actually got a chimeric mouse and a black mouse. Now you take the chimeric mouse and you actually back cross with a black mouse where you have all X plus. So think about that. These will actually basically all the different alleles. This will be the allele from the black mouse. So this is basically from the black mouse. These are derived from this line. So now if by this black cross, if you get back the brown mouse, all of these are the progeny from the ES derived stem cells, because that's basically because they are not chimeric, they are not black. And you screen the brown progeny for the DNA to identify the X minus right here, X minus and X plus heterozygote. So that's what you do. And then you screen the progeny for the DNA to identify the X minus and the X plus homozygous as well. So now this is how you can actually get a knockout mouse, a very intelligent way that you can actually get a dominant negative allele. So how this in reality works? So suppose here what you are seeing that this is an inactivation of the function of a wild type GTPase by the action of a dominant negative mutant allele. So the dominant negative allele can be inhibited by this process. So what you are seeing here, a guanosine nucleotide exchange factor, which is GEF, activates a small monomeric GTPase, and by catalyzing the exchange of GDP for GTP. So when this is functional, the guanosine nucleotide exchange factor, GDP will be replaced by the GTP. So this is the process. So GTP actually can bind, and these are the active form of the protein. Now you have a cell which is expressing both wild type alleles and a dominant negative allele. So what is happening in this process? All these are produced. GDP is bound to that. But in this protein right here, in a cell where you have the guanine nucleotide exchange factor, this dominant negative mutant is going to actually block the site. So all these cells which are actually producing this GTPase, they will not be able to use this site so that it can be exchanged to GTP. So this is basically the endogenous wild type GTPases become trapped into the inactive GDP bound stage, which is phenotypically similar to the homozygous carrying the two recessive GTPases loss of function alleles. So this is basically a knockout by a dominant negative allele. So this cells is actually producing that, but its function is knocked out by a different allele. You are not really knocking out the gene as such. So this is also another way that you can silence these genes. So the next one that what we are looking at is another way that RNA interference or RNAi that also can inhibit the gene function. So what happens that this is actually a process by which the double-stranded RNA is made into a 23 nucleotide RNA called a siRNA. These are the non-coding sequences, and these are produced in the cell as endogenous RNAs called microRNA silencing RNAs, and these are actually involved in the regulation of gene expression. These are produced. So we actually take the advantage of this, do a specific type of gene silencing. So how you actually do that? There are two processes. Let's take a look at if we just actually do an in vitro production of this double-stranded RNA, how we actually can do that. So suppose you actually have a gene of interest, and you can actually produce a transcript which will actually have this sequence right here, 5' to 3'. Suppose the letters are sense. This is the amino acid. I mean, like, this is just a hypothetical, but think about that. It is actually making sense right here, which will be one strand of the double-stranded RNA. Then what you do that you actually have another construct where the gene is backwards. So the sense is from the backwards. So this is basically the complementary strand. So here you are just making only one strand, 5' to 3'. Here you are making the 5' to 3', but it's a complementary strand. So it will actually flip and it will make a double-stranded RNA, which is taking the mRNA from here, mRNA from here. So what happens that the dicer that we talked about, that this dicer will actually make it in a 23 nucleotide RNA, which is called a siRNA. So this is an in vitro production of this process, and we can actually utilize this to silence the gene. So here you can see that if you inject these into the embryos of, say, Caenorhabditis elegans, which is a worm, here is actually a reporter gene which actually will fluoresce. So you can see that this is actually a signal where the gene silencing did not occur, whereas here the silencing completely occurred because the reporter gene actually did not produce any color. There is also another way that you can actually do this process in vivo. That means in the animal, you can actually do this double-stranded RNA. So how you do this in this process, what you have, that you actually do have a tandem sequence, and you actually inject this construct into the target choice. So only the difference is that here you have to make a sequence where you have a tandem sequence of sense. What do you mean by this? You have this sense, that is the sequence. Then you need a couple of different gaps. So you need a couple of different nucleotides right here. And then you put the sequence again in sense. So why this is required, we're going to come to that in a second. But suppose this mRNA is actually being produced. So what will happen that as you have this tandem sequence in there, this will actually make an hairpin. Think about that if this actually is the sequence right here, then right here, these sequences which are joining the two sequences are in there. And here the sense sequence is from backward. So this will be the antisense of this. So it will form a double-stranded RNA. So when you actually look at that, because see that from E, this S will actually come over here. This E will come over here. So because it is actually hairpinning like this way. So just remember that you have tandem sequences, automatically this will be an antisense. So once you actually have that, this is a short hairpin RNA will automatically produce in the cell, and dicer will actually cleave that hairpin and form the siRNA. So this process, what we are seeing, that suppose you have the double-stranded RNA or the hairpin that we talked about, and the dicer will actually cut it into a 23 nucleotide sequence, which is a siRNA duplex. Then you have a step where you actually form something which is called a RISC, which is an RNA-induced loading complex. So this actually contains a protein called Argonaute, TRBP, transactivation response element RNA binding protein. So you can see that this is actually a RISC complex with a dicer, TRBP, Argonaute, and the double-stranded RNA. But just have an overview of that this RISC complex is going to remove one of the strand, and now this RNA is just single strand. It will remove the double-stranded RNA, one of the strand. So now what will happen, that suppose this RNA, the silencing RNA, actually is complementary to a specific mRNA. So now what will happen, that specific mRNA will now be degraded by this RISC complex. So this is the process of silencing step. So why these RNAs are actually produced in the cell, which are complementary to specific RNA sequences or mRNA sequences in the cell. This is a process that where the microRNAs can actually silence or regulate the gene expression. And the advantage of this is that you can actually design this this RNA wherever you want, which has to be complement to a gene sequence. And if you just insert that with the help of this the RNA silencing complex, you can target a specific gene and you can silence that gene. So the next thing what we are going to talk about is the CRISPR-Cas9 mediated genome editing. And in this process, and the CRISPR actually stand for clustered, regularly interspaced, short palindromic repeats. So what we mean by this, that you have short sequences which actually read forward and backward the same. So those are the palindromic repeats. And these were actually, the system is actually discovered in bacteria, where they actually use this system to disrupt those phage DNAs. So this was actually a process, and this is a primitive process kind of conferring a resistance against the infecting phages. And this is actually utilized, and we have actually seen that this actually works in almost all mammalian systems. So how this actually works? You need to have a specific RNA sequence, we call it a guide RNA, that will actually attach itself with the target sequence that you actually want to edit. And that specific gene sequence will then be disrupted by this Cas9 nuclease. So you actually target the guide RNA to the sequence you want to modify or delete or want to edit. And then you actually use the Cas9 endonuclease to do that function. So let's take a look at how you actually generate this system. So first, you have to have a guide RNA expression. So how you actually do this, you actually have a target sequence, and you have a species-specific promoter, whether it's a mouse, whether it's a zebrafish, whether it's a Caenorhabditis elegans worm, or human cells. And right here, there is a sequence which you call as a track RNA scaffold. This is actually needed to bind it the Cas9 nuclease. But this is the most important part. This is the target sequence, and this is actually the sequence that you are trying to either delete, disrupt, or want to edit. This is the most important thing. And this is almost the similar for the Cas9 system. On the other hand, what you have, that you actually have a codon optimized Cas9 gene. What we mean by that, see the codon usage, that the 3-letter codons that is used by the bacteria are a little bit different compared to the preference is a little bit different from the mammalian codon choices. So it does not mean that the genetic code is different. So these are codon optimized for either the specific mammalian system, any different vertebrate, fish, amphibia, any other cell type, and you have a species-specific promoter to that. And then what you have, that this will actually produce a functional chimeric guide RNA, which is attached to this scaffold. And this will produce this Cas9 nuclease. Now, once you have that, what you have right here, you have the genomic DNA target sequence right here. And this is the gene of interest. And you have actually made this target sequence right here. And you actually take just about 20 nucleotide target sequence, and this is your target sequence. So what happens that this red part, as you can see, that this red part will actually, with the help of Cas9, will actually right here hybridize with this yellow part. So this complex will actually make the 2 strands separate, and this RNA will actually hybridize with the DNA, and the Cas nuclease will actually cleave this site out. So this is basically the process of targeting the specific sequence. So you can see here, there are 2 ways that you can do. You can just take a small sequence and disrupt that gene, and it will break. So just the deletion of a few base pair. And now you have a non-homologous end joining. So you have a short deletion in the gene. And if that happens like this, what you will have, that particular mRNA will have a complete mutation or a frame shift mutation. It will probably stop at a stop codon earlier found in that, and that gene is now silenced because none of these mRNAs will produce any functional protein. So this is just the CRISPR-Cas9, just to delete a few sequence, to disrupt the gene sequence, to make a mutation, non-functional mutation. This is one process. On the other hand, what you can do, this is another powerful technique, what I mean by editing, you can actually make this specific sequence of your guide RNA, and now you actually have this flanking region of the gene right here, these 2 yellow part, and along with that, you actually do have another template where you actually design a sequence, a specific change in the sequence, which could be almost as 100 nucleotides, and having the flanking region right here. So what you actually do in this process, when you transfect the cell lines or the animal with both of these, you will have this homologous recombination. That means this specific portion can actually go into the gene and taking that black edited part, which could be as long as 100 base pair long, into the gene. And this way, that although you cannot do a very large portion at a time, but you can constantly change, and you can edit these genes however you wish by actually changing this RNA sequences right here in the end. That will specifically make these, so changing in the DNA sequence will insert themselves by the homologous recombination to form a mRNA that is now completely edited. So this is the very powerful technique of the CRISPR-Cas9 mediated genome editing. So here in this process, you can actually see that this mouse actually do have a mutation in the CRY-GC right here, which actually produce a defective gamma crystalline protein, and that actually cause cataract. So this is already a germline mutation. But what happens using a CRISPR-Cas9 system, these gene is actually repaired, and this is the recombinant mouse, which actually doesn't have that So this has actually been cured. So you can easily see that how this CRISPR-Cas9 mediated correction or genome editing can actually have an impact. So this is a classic example of this. So this is basically the end of the part 5, and that will be basically the end of the module 1.1.